Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really fascinating guest today involved in research that's helping to create a better tomorrow on so many unique fronts. Today, we have the honor of being joined by Dr. Jakob Hanna, who is senior scientist and professor in the Department of Molecular Genetics at the Weizmann Institute of Science in Israel, where his lab and the interdisciplinary group of scientists within it are focused on understanding the complexity of early embryonic stem cell biology and early developmental dynamics, as well as advancing various areas of human disease modeling in the laboratory. More specifically, the lab is focused on investigating the detailed processes of cellular reprogramming, in which induced pluripotent stem cells can be generated from somatic cells, and they ultimately investigate how this pluripotency is maintained throughout development, studying both mouse and human models. In their studies, they employ a diverse arsenal of biologic experimentation models, including high-throughput screening, advanced microscopy, genomic analyses, ultimately seeking to combine biologic experimentation with computational biology, theory and modeling to really elucidate these various biologic questions. Dr. Hanna completed both his medical degree and his Ph.D. At the Hebrew University of Jerusalem. His thesis was focused in that work on the domain of immunology, looking at interesting novel functional and molecular properties of human natural killer cells. Then he went on to do his postdoc studies at the Whitehead Institute of Biomedical Research at MIT under none other than the tutelage of Professor Rudolf Jaenisch, studying pluripotency and epigenetic reprogramming. When he's not doing all of that, Dr. Hanna is also the founder and chief scientific officer of a new biotech company called Renewal Bio, looking to leverage the power of these new stem cell technologies, potentially applying them to a wide range of human ailments, including infertility, genetic diseases, potentially longevity and aging. We're honored to have him with us today. Dr. Jakob Hanna, thank you so much for taking the time to come on the show.
Well, thank you first for the kind introduction and for having me and for the interest in this topic. Glad to be here.
It's a fascinating set of topics. I'm really looking to get into all of this with you. Jakob, I would really like to start off at the beginning a little bit on about how you got interested in this field. And then also, I found it very intriguing, sort of looking at your PhD, which was also in another area of sort of retasking cells. In that case, you were studying natural killer cells, which we normally think about in sort of the immune system and fighting infection and preventing tumors. And you were studying, hey, there's also potential here, modulating these cells that they do other things in terms of placental tissue development, helping us reduce transplant rejection, other applications. I thought just this general theme of reprogramming, retasking human cells is just so fascinating. Talk about how you got started in it, if you would.
Yeah, I mean, it is an interesting way of looking at it. And I did my PhD on how natural killer cells, which are immune cells, actually, particularly in humans, natural killer cells are typically actually very aggressive immune cells that attack virus-infected and cancer cells in our body. But in pregnancy, actually, they can reach about 40% of the cells on the maternal side of the placenta. And there was a big question, what are they doing? What could they be doing? And indeed, we were showing that they actually have a role in modulating the tissue and even promoting its growth. And kind of, we call it at the time, a time to kill, a time to build.
Mhm.
And then, when I actually went to do my postdoc, I did not continue in immunology. I wanted to go into stem cells, which now it's funny because I turned, to the other side of the birth. I was looking from the mother side. Now I'm looking from the embryo side. So maybe, and as we'll talk perhaps today, we're also going back to immunology and then taking immune cells and transplanting them. So maybe there is a circle here where, we are closing after all, we're having this kind of full different angles on the fetal maternal interface and the fetus itself and the immune systems. So perhaps they are connected in the end.
Yeah, absolutely. I've sort of actually, be interested before we get into sort of the details of some of what you're doing. I thought we'd actually take a little historic walk here because you are part of what I'll call a very unique lineage of science here. And if you just look, because you're there in Israel, I'm sitting here on the East Coast of the United States in Philadelphia, and you did your postdoc with Rudy Jaenisch, and probably a lot of people don't remember that Rudy 50 years ago was here in Philadelphia, starting his career off with another local luminary, Dr. Beatrice Mintz, working on sort of the first transgenic animals. And then 25 years prior to that, you had really the experiment that I think about that started all this stuff off, the Briggs and King cloning work, 1952, same facility, Fox Chase Cancer Center. This leads forward to John Gurdon's education in oocytes and ooplasm. Fast forward, you get to Yamanaka a few decades later, and then ultimately Jakob Hanna. You're taking things forward from here. I think the interesting thing here, I'd like to say a few words about, is that whether we're talking about human development, regeneration, some of the more exotic things that Mintz did in the area of cancer reversion, at the end of the day, embryology, embryo sciences is behind it all. Talk a little bit about this, if you would, because I think we don't think as much. We sort of think about embryology as this thing that's sitting over here. We make babies and that's it. But it really feeds into a lot of what we have going on today in some of these areas in 2022. Talk a little bit about the history, if you would.
Yeah, I think, there are a lot of questions in biology and perhaps might sound a cliche, but really, I think, in the end, I think it's the most amazing question, how every entity or embryo starts with a single cell. It's a very, very fascinating field. It's also shows that there's a lot of power that is unleashed basically during this process, and we would like to understand it. I think that's what starts there, and then you start getting a lot of layers because you start thinking the field of epigenetics start there, which is a lot of and there's anyways a lot of epigenetic drama happening there about DNA methylation and remethylation, X chromosome inactivation, which are happening there and really happening in very drastic and has critical influence. So it became also a system that is for modeling these events, for understanding these events. It's like a very fertile ground, whether you're looking about single cell technologies to apply them in this model, or you're talking about microscopy, apply it in this model, because it's a very relevant question and it has a lot of information. And then when you have then the genetic engineering, because well that's the start of the embryos. If you want to engineer an embryo, so that's where you start. So it's all indeed it comes back a lot of these events. And again, yes, and we talk about the touch with cancer. Of course, there's a lot of the events of epithelial to mesenchymal transplantations, which became relevant in cancer. Changes in DNA methylation, actually, many times in cancer, some of them are very hypermethylated. What does that mean, and how is this different, similar to what's happening in the embryo? If you talk about oncogenes and retroviruses, which are also sometimes being regulated carefully and during embryo development. So it really becomes a very kind of fascinating model system to look at these very key events. And then, of course, in addition, once you start growing these cells in the Petri dish, that kind of entered another layer that became interesting, because both they become a very good model to look at, as a model system, they are similar to what's happening in vivo. So things that are hard to do in vivo, like biochemistry or screening, at least you can start with ES cells and then go back and look in vivo. But in themselves, in vitro culture stem cells have a great potential application for transplantation. So that opened another area there. And So definitely, it's a very central, I call it, it's a very busy neighborhood.
Absolutely. Absolutely. So, Jakob, looking at, sort of the different areas that your lab is currently focusing on, one of them, that's highlighted first and foremost on your lab's webpage is sort of this general area of deciphering cellular reprogramming. And, you published this very interesting article a couple of years ago, saying in 2014. It was entitled "Hijacked by a Oocyte: Hierarchical Molecular Changes in Somatic Cell Nuclear Transfer." And you sort of go through the history, starting with John Gurdon and a little bit of sort of where we've come with this complex hierarchical processes of, all these transcriptional and epigenetic dynamics. Decades later, we come to Yamanaka and his factors. And in between, there was a lot going on in those decades. You've been studying, you've been going a little deeper, sort of looking at different regulators of chromatin, looking at sort of what else is essential as we think about cellular reprogramming, which is an extremely complex process. Talk a little bit about some of the work that you're involved there, because I know that, you think a lot beyond just, okay, these 4 factors. There's a lot more going on that is involved in retasking cells. Talk a little bit about your work in sort of unpacking cellular reprogramming that's going on in your lab.
I think, very much we're driven by curiosity, and sometimes even you could call them, philosophical questions, which we never got to answer, but at least they're there. And one of them, when you talk about the Yamanaka experiment or the cloning, is like, why does it work? Why can you take a cell and turn it all the way back to pluripotency? And I would say there were you could think of like 2 paradigms. One is like saying, well, every cell has a transcription factor code, and if you have the right transcription factors, you can induce that state. And there's some evidence supporting that. So like experiments, if you express, like in MyoD, you can get fibroblasts to become like a muscle-like and so forth. Or there's another, and we'll talk about it, and then there's another option, is that pluripotency is actually a very unique state. It's not just another cell type, but it's actually a state of lack of identity, meaning it's like you're erasing everything, and that is the default state that comes up. And that is conceptual. I think there's a very 2 things. And I actually now more and more, our research is like teaching us that it's actually the latter scenario. That the reason that this works, that pluripotency is a state of lack of identity, that you tend to erase epigenetic marks so strongly, in the end, you awaken the endogenous program, and this is a very stable state. And I can explain more. Versus, if you look at, if you go about the what I call the transcription code paradigm, well, you can never, if you try to get a fibroblast to a neuron or a fibroblast to a muscle cell, you can get muscle-like cells or neuron-like cells, but they're about 30% like. They're never almost, not even close to 100%. And I think that is something that is obscure. Therefore, a lot of the research we were studying is like, okay, so there's something very unique about ES cells. And that's why, as I said, it's lack of identity, which means they don't need to repress. So there's no need for epigenetic repression. And that is why we started getting into epigenetic mechanisms, and particularly looking at mechanisms that are repressors. What are the role and the importance of these repressors, and at what stage and what they do? And that's why a little bit kind of led us on some of the work we started. The first paper, which we are, at the technical level, they would go, okay, this enzyme, and we knock it out, and it does this, and it does that, and then at this stage, but and you have like 3 or 4 or 5 papers, each one looking at a different enzyme. But I think if we put them together, that was the motivation for looking at this. And really the conclusion for us was that indeed, to make pluripotent cells, you need to remove repressive machinery. And not only that, actually, pluripotent cells, once they're established, they no longer need this repression. So they don't need actually DNA methylation, which is repressive. They don't need microRNAs, which is repressive. They don't need RNA methylation. They don't need polycomb. the cell is stable. They need them as they progress towards differentiation.
Mm-hmm.
So that is so, and that's in a way, if you want to think about this again, kind of conceptually a little bit, you could talk very philosophical or blah blah, but you might think that the early stem cells, and we'll talk then about what I mean by early stem cells, is that in a way perhaps it's akin to our yeast ancestor, because like
Sure.
yeast don't have DNA methylation, they have RNA most of their life, they don't have RNA methylation, so they're like, so in a way the stem cells actually are very kind of this basal basic state that doesn't need a lot of these fancy mechanisms that came later, but they can, of course, they become important for differentiation.
Mm-hmm. Mm-hmm.
But as we started in parallel looking at these mechanisms, we started asking, well, there are different type of stem cells. There are ones that correspond in the mouse corresponding to the blastocyst, which is about day 3, and then to the ones that a little bit later, which correspond to day 5, which we call them naive pluripotent cells and prime pluripotent cells. And at the time, the feeling in the field was, well, yeah, they're quite the same. These are flat, these are domed. It doesn't really matter. They're both stem cells. But as we dug deeper in this, we also realized actually, no, they're actually very, very different. So, the property that I talked about, that naive pluripotent cells can tolerate loss of repressive mechanisms, this doesn't hold true for primed cells.
Mm-hmm.
So primed cells are behaving like differentiated cells. They need these mechanisms. So they're very, very different. And this is a way kind of surprising, because we showed in a lot of cases, you're talking about that actually they're completely opposite. They respond completely the opposite way if we remove DNA methylation or RNA methylation, and really putting that, I think, for the first time, that these cells are very, very drastically different.
Mm-hmm.
And then that going there, expanding, that allowed us to start asking what's happening in human cells. Because human cells, in the end, as we showed in others, they're actually the ones that are conventional ones that were there for many things are more primed cells. They do not tolerate loss of epigenetic repression, and they are more similar in trait to the mouse primed cells. And that has, this kind of ideas pushed us to ask, well, can we get naive pluripotent cells in humans, and what does naive cells mean? And it's a very hard thing to test for, because you don't have a functional assay in humans. In mouse, typically, if you want to talk about naive cells, and going back to the Jaenisch and Beatrice Mintz, you get the ES cells, you inject them, you make a chimera, a mouse is born. That is, of course, it's unethical, illegal to do this in humans.
Sure.
But we actually, that line of thinking led us to think, okay, maybe we have a quality, which is, can we make human cells that can survive without epigenetic repression? And indeed, last year, we published a study where we screened for conditions, and indeed, we can isolate human cells that are indeed naive cells. They can tolerate, like the mouse conceptually, they can survive without microRNAs, without DNA methylation, RNA methylation. So that has, in the end, this line of thinking allowed us to get different pluripotent states in humans. And then that has brought us to the really most exciting question, which is, why across different mammalian species do we have different types of pluripotency? Why not have a binary situation, which is 0 versus 1?
Right.
Why the cell has to kind of go from naive and not directly differentiate? It kind of goes into a slow decay towards primed stem cells. They're still stem cells, different properties, and then differentiation happens. So what is the consequence for that? And although we don't have the answer, but then we actually then show that this matters, particularly in humans. For example, if you want to make germ cells, which are the progenitors of sperm and egg, you have to start with a naive pluripotent cells in this ones. In mice, for example, you start with naive cells, you have to give them priming like in development for 2 days, and then you make PGCs. If you don't give priming, you will not get PGCs. If you do this priming for too long, then you will not get PGCs either. So it kind of, if you think about it, it shows you that there's a window of competence. You have to start with this most potential naive cells. You have to open up slowly the situation, and you have a window for big competence to make PGCs. So that is at least showed us functionally that the cell type of stem cells matter to get the function. And that is now the challenge, I think, for us, which is really understand molecularly how is competence or lack of competence established. So now we have the phenomenon very calibrated, but what is the molecular basis? And then is there a paradigm that is happening to all lineages and not only germ cells? Does recapitulating the sequence of events that happen like in the embryo, because the embryo, again, you start with naive cells, you have then primed stem cells, and then you get overt differentiation. And again, we haven't answered that yet, because we didn't really have a good system to answer that. Because if I want, what is the readout of a naive stem cell becoming a primed stem cell? The readout is that continuing and making an embryo from those stem cells. Right. That is the readout. Right. And if I want to make a mutation, if I want to change something, this is great. I can easily do it on the stem cells, but we cannot make directly from them whole embryos and look at the phenotype, which is what is needed to answer this question. And that is why basically this has led us to what we call now synthetic embryos, or a better word is actually stem cell derived embryo models, which we call SCMs. And this is why we started doing them. But then, again, if you are trying to grow an embryo outside the uterus from stem cells, then you ask this question, well, how on earth are you going to do that if you cannot grow natural embryos outside the uterus when you take them out? And that is why, we had to wait a little bit and go develop a platform to answer this question, which is, okay, can we grow embryos after implantation and watch them do gastrulation and organogenesis? And if we can do this, of natural embryos, which ones are basically, we took them outside the uterus. Once we define the platform and conditions that can support natural embryos, then we can move on and take in vitro plate cultured stem cells and put them in these conditions and see if then they can make embryos, and then analyze these embryos models. And that has been basically our journey a little bit, and we're happy with this. If you look at our papers, as I mentioned, some of them are looking at epigenetic repression, some of them are looking at naive pluripotency, and some of them are looking at germ cells, and some of them are looking at growing natural embryos or making stem cell embryo models. But there is a very line of thought or platform development to be able to answer these questions, because these are very, very connected. Perhaps every paper puts a spotlight on different part of that neighborhood, but we're trying to kind of, now we feel that our things are coming together, and these platforms and these questions are feeding in each other to advance the technology and also answer the biological question.
Outstanding. And Jacob, I appreciate you walking us through that. So you've taken us from the naive and the prime cells into chimerism, and then now one step beyond, and obviously the seminal paper from last year, post-gastrulation synthetic embryos generated ex-utero from mouse naive ESCs. Could you talk? I mean, because there's really, there's two, as you were saying, there's two major things. Well, I mean, there's a lot of major stuff going here. One, mechanical womb technology for allowing this to happen. But at the same point, that this is the general concept of a synthetic embryo, which I knew, when this paper was published, obviously the press ran in all directions with it. Talk just in general, if you would, introduce us to a synthetic embryo for a minute, because we basically have, the way I think about it, we normally have sort of the cellular stuff going on that creates the body, the sac, and the placenta. now this is happening sort of in a stew, let's say.
Yeah.
Introduce us in the audience to synthetic embryos as if we didn't read that paper or we didn't know
Yeah.
about the publication.
Yeah. No, definitely. I think, in general, this field is now, like the name is what's called stembryology, which is
Yes.
making embryo models from stem cells. So it goes to embryology.
Yeah.
And there's been, a lot of interested thought and recent progress in different fields, which is the idea. So we know that we grow stem cells in a dish, right? That's the concept. And we know if we inject them into another embryo and put them inside the uterus, they can contribute to that embryo.
Right.
So they have the potential. And the challenge was, well, can we make the entire embryo only from stem cells? Okay. And then you started having different models. One of the first model, I think, that had all three lineages of the early embryo. So the three lineages will be the embryo proper, the primitive endoderm, which make the yolk sac, and the trophectoderm, which makes the trophoblast of the placenta. And the first model really that had all three lineages was a model from Nicholas Riveron that he made blastoids, where he combined ES cells with trophoblast stem cells and grew them for 2 days, and the ES cells gave rise to the PRE, and he got structures like blastocysts, and he showed that you can look at some interaction, some adherence, and it was an early model. However, there was this kind of question, is this kind of a bona fide embryo really, or embryo-like? Because so typically you have it for a natural blastocyst, you can put it inside the uterus and it will grow. And when they planted them, they couldn't grow. They did not develop, not even for 1 day further. So that was kind of a bit, shows the limit that this model is actually something is not authentic about it, which is still we don't understand why, which is very, very puzzling about it. And then and then you have models, for example, like trying to make gastroloids, which is like ES cells, and you give them like a 1-2 pulse of wind, and then you can start get this kind of axis formation. Even you can get somites if you give like matrigel, which is like adhesion properties in the media. These are other types of models that happen, and there are other ones that are developing. And we basically, what we did a bit was different, and which we said, based on different observations we had, actually mostly with human, actually it began, is that we noted that human naive cells, for example, can very easily become trophoblast stem cells and can very easily become primitive endoderm cells, which is perhaps not surprising because that is their natural progression.
Yeah.
But the fact that they gave trophoblast stem cells, it was surprising because the thought was always that that potential has been lost, that the segregation has happened, whatever is going to become the embryo, whatever is going to become the placenta is already separated. And that's why we started thinking, well, when we're trying to do in mice, which is what we did, let's try and do everything from naive pluripotent cells when we're going from the mouse, which I think, is, I think, like one of the, as I agree with you, I think there are two exciting things, at least to me, in the recent paper, not only making to a really bona fide organ-filled synthetic embryo models, but also that we did everything from naive ES cells.
Yeah. I
think that is another point, because as you said, because it shows you at the end that naive pluripotency is basically totipotent, that if you perhaps tweak it with the right signaling, you can still have that potential and give rise to the two other lineages, and everything comes from there. And then as I mentioned, the bottleneck was what is the problem? So other scientists, for example, made aggregates of ES cells and trophoblasts, and they got embryos of about day 5, day 5.5, but they could never evaluate further because in utero transfer, to put back, you can only do that with the blastocyst stage. If you have a natural day 5 embryo, once you take it out of the uterus, you cannot put it back. And I'm talking about the natural embryo. So there was no way to test the ability of these embryos.
Yeah.
But once we developed, which we published last year, a platform to grow natural embryos, and I emphasize from pre-implantation, which was never done before, from day 5.5, and capture the entire gastrulation, and those gastrulation embryos could even complete organogenesis. So we can go from day 5 to 11.5 with natural embryos. So now you had a system to test. Now those claims could be tested.
Yep.
Because I think a lot of scientists were getting away. We're making, this looks like an embryo, but with all important study, but a clump of cells that makes a lumen is not a really proof that this is an embryo.
Right.
And indeed, actually, a lot of these embryo model that were published and they look, like elongated with a lumen, when we put them to the test, they failed. Okay, so we actually had to revisit which cells we should aggregate, and what is the media we should aggregate them in. And then when we revisited that and really found new conditions, and then we subjected them to the conditions we defined, which we know can support a natural embryo. Mm-hmm. So indeed, and that worked. And this is the recent paper. In the end, once we solved the bottleneck of how to go from pre-gastrulation to late organogenesis with natural embryos, then we said, okay, we can now test and take stem cells and perturb them and put them in these conditions and watch, can they reconstitute an embryo-like entity, which is what we call a synthetic embryo or a stem cell-derived embryo model. And this is what we got. We showed that, yes, we can get embryos models until day 8.5, which means they completed gastrulation entirely, made all organ progenitors, and even started organogenesis, including heartbeat with valves, with chambers, and neural folds, neural tubes, somites, gut tube, tail bud, neural crest, PG, primordial germ cells, including their migration. And we can, and the interesting thing is that we can do that also by starting from naive stem cells only. And that is what the journey has led us. And going back to what I told you before, although the system at the moment is inefficient, once we make it efficient, then we will have what I told you before, which is we start with naive cells, they go priming, they make an embryo model. I have the platform I'm after. So now we have it, the efficiency is low. So we need to get the efficiency higher to be able to really then make a mutation and watch the same embryo, how this mutation perturbation unfolds through different stages of development. And that is what we are after, at least one of the major goals.
It's completely fascinating, and I'm sure you'll increase that efficiency, as you continue to work on this. And clearly, just listening to you, and obviously looking at this paper and looking at the applications here, in terms of studying everything from genetic issues in the early embryo to, of course, the potential leveraging of these various cell lineages in terms of organogenesis. There's so much potential here. Obviously, talk about the science you're doing next and in improving this. Let's talk about the future visions here in terms of renewable bio, because I think it was whatever the MIT or the Harvard, whatever the press was, like, hey, well, looking to create individualized embryos to produce potential stem cell lines and so forth. Take us a little bit into the future, what you see there, because this goes in so many directions. I'd love to hear it straight from you.
I think actually, as with iPS cells and like with a lot of technologies, I think first and foremost, we want to understand the mechanisms of this.
Sure.
The reasons for, how does really the cells sort out and or find themselves into this entity and start making these synthetic embryo models, stem cell derived, and that will eventually lead to improving the efficiency, improving the quality and authenticity of the model. Because once we start these defects, what is needed, then we can compensate for that. And also, it's very, we are human beings. We're almost, most and foremost curiosity driven. So it's very interesting to look at these effects. And then I think so that is, a lot of the questions. For example, why with natural embryos, when we grow them outside the uterus, can we reach day 11? Actually, we can now reach day 13 in the lab. We haven't published it. Versus with these embryo models, we can reach so far only day 8.5. Mm-hmm. So a lot of these really interesting questions, and this is just the start. I think the field is going to boom further because, now once you can, now you can, you have more faith to really invest more resources, more time, and go further. And of course, then, it brings us to look at other species, because we want to, before we get to humans, even if you think about monkeys or even elephants, okay, it's very hard to study embryos in these models because you can't really get embryos, and it's very difficult, very expensive, a lot of ethical challenges. And this has been limiting our knowledge to look at embryology in different species. And we need to do that. The mouse is great, but the mouse is actually an outlier in a lot of things when it comes to embryology. And I think that is another very fascinating event when we try to go with X-utero embryogenesis, whether we're talking about natural or stem cell derived CMs. For example, we're looking at the rabbit, because the rabbit, actually, its shape is very similar to the human. And how does gastrulation happen in the rabbit? Of course, rabbits are more approachable, they're very fertile. So that is as a model, and it's a model, it's a very interesting one. And then going to we're working on monkeys, we're working on pigs as well, embryo models, and now we can do that. And then third, of course, embryo models of humans. And it's really an understatement when we say human gastrulation is a black box, because we have absolutely no idea, because we cannot get access to such material. It's actually almost unethical to do it, because usually so you're talking about days 14 to 28 of pregnancy, and usually the woman doesn't know she's pregnant, even if she agrees to do an abortion. And then why would you do what's called a curettage, and when she can get a pill and just end the pregnancy, which is much safer. And even if somehow the abortion happens for some reason, the embryo, you get one sample, probably the embryo is really distorted from the process itself. We cannot get this material, basically. And not only that, as I told you, embryology, if you want to study one thing, you don't want, we take what we can, right? But ideally, you don't want just like a snapshot of different samples. What you would like is, watch it in your own eyes and see this unfold, particularly about gastrulation, which is very dynamic process, and migration, and physical and mechanical. And that's why, if we think, if we can take naive stem cells in humans, which fortunately, as I mentioned, now it pays off because we already have conditions to grow naive stem cells for a lot of reasons. So that obstacle has been solved. And we know they can make trophoblast stem cells and primitive endoderm cells. So that has been solved as well. Then can we make early embryo models in humans just from stem cells with, of course, with the approvals, with the informed consent of the donor that is doing that, which is very important, and start seeing how does gastrulation happen in humans, and start analyzing this genetically, meaning, which pathways you can make a mutation, and just look how does gastrulation happen, and really learn what are the key pathways to make the organ progenitor. So I think, if we look at knowledge of development, which for sure will unfold into knowledge about the origin of defects in pregnancy, of embryo defects, I think we will learn in the end, right? We want to differentiate stem cells in the petri dish into different cell types. We want to mimic the embryo, but we don't know what's happening in the embryo. So, if we learn about what's happening and what are the cytokines that are important, maybe we can also copy these principles and then apply them into conventional 2D differentiation of stem cells into a certain lineage. But then, then this is where I guess perhaps, our research, and also in Renewable Bio, the company that we're doing, this kind of opened another concept, which is, if you think, again, going a little bit, philosophical, blah, blah, I call it level.
No, I like blah, blah, blah.
What our experiment showed, the two papers from last 2 years show what they show, basically. One is that, okay, I told you we can take an embryo in the mouse from pre-gastrulation day 5 and grow it until day 11. What this shows that the uterus is not dictating just shape of the embryo. It's not giving the morphogens, which give the which give the shape of the embryo. It's about metabolic support, which we can substitute. So the embryo is it's a proof that the embryo is self-organizing, it's self-propelling, it's developing. And then the second paper takes it to the next level, which shows actually that naive stem cells have all the capacity and all the information encoded within them that allow them to unfold into self-organizing embryo models, which are self-organizing by nature to grow further. And then basically, if I go back to another major problem we have in the field, and I think, if we keep, we can a little bit, the field, if I want to say, like, we cannot hide our head in the sand any longer. We are having a major difficulty in differentiating into a lot of cell types in the petri dish. It's actually, if you think about it, 20 years ago, if you would tell people, actually going backwards and making self-iPS stem cells is going to be solved before differentiation, people will say, oh, no way. But here we are.
Right.
And that actually is proven very, very difficult task. And it could be, of course, there's a lot of successes, and I'm not dismissing anything in general, but I'm just saying there is a challenge. I mean, look at it, even hematopoietic stem cells, even in mice, nobody for 40 years has been able to make authentic hematopoietic stem cells in mice or in humans that can transplant into adult mouse. Versus, if you take these stem cells and you make a chimera, they will make a bone marrow in that chimera. Yeah. So that shows you that the cells are fine. We are not able to recapitulate this outside an embryo context. And it could be that if you think about how do we envision differentiation inside an embryo, it's a sequence of many events that need to happen. If we talked about windows of competence before in the germ cells, you can think of, there are a lot of windows of competence that need to happen. You need different cytokines secreted at a certain magnitude at a certain time point, that if they happen too early or too late, this is not good for the quality or efficiency. You have mechanical factors. It's way complex. And it could be for a lot of lineages, it's just too complex for us to come and dictate the morphogens and the mechanical cues. And then since we are able to get something similar to an embryo model out of stem cells alone, and again here, we are not giving morphogens. The embryo is secreting the winds.
Right.
The embryo is making the axis. The embryo is dictating things. And basically we say, well, maybe the solution is the self-organization, since we do not have to recapitulate these very refined sequence of events that need to happen by us inducing the morphogens and differentiation cues. Let the stem cells roll into becoming an embryo model, and then actually use this, we kind of hitchhike on this process and take the lineage progenitors that happen on day 30, day 40, day 50, and then either use them for transplantation or expand them further and use them as a starter material for these conventional 2D differentiation protocols. So that's why, it's not only about mimicking the embryo model, but maybe actually we can make an embryo model to get and look at it as a very sophisticated and authentic differentiation protocol. And I think that is where my lab and Renewal Bio is trying to look at it. And I think, again, I strongly believe that you need a holistic view of things. As I mentioned, you need to understand the mechanisms to improve the efficiency. You need to learn the ins and outs of different species and compare them. And I think that will feed into improving the protocols on human cells. And it's a kind of a multi-layered effort. And I think it's beyond the scope of define one lab, and that's why we're doing two things. I think we are providing our platform basically to every academic institution based on basic MTA to be able, because a lot of things to be done. By the way, all the information on our protocol website, how to get everything, how to sign everything, we really would encourage people to do that. The second is, industry is important because, they can have the money to do a lot of experiments. What we did with the enhanced incubator, it's a condition. We're not saying this is the best condition. This is not saying this is the only condition. Probably there are other ways to do this. And we want to systematically look at this. And so this is a very big endeavor. And I think that is, we would like, help of bioengineers and budgets and really doing this, automating this and trying to predict which ones are the good ones to kind of do this, and that is why the effort that is being done here. But what I do want to emphasize is that it's not our goal, and we cannot make what we call people imagine they hear of this, we're trying to make embryo models, that you're trying to make embryo model to grow an embryo outside the uterus and make a pregnancy entirely outside the uterus. Right. So I don't think we can ever really make a pregnancy of a human being for 9 months outside the uterus. We're not trying. I think we're not trying to put them into a uterus. It's illegal to put it into any uterus. That is not the goal. Yep. The thinking about this is actually about, it's an early induction protocol, differentiation protocol. Yep. You can look at it as a universal induction, which means you start with all progenitor cells and then you go and select what you need. And this is more about the scenario we're looking is about patients who are looking for cells for transplantation. Mm-hmm. That is what we're trying to get at, and how this can benefit them.
Absolutely. Yeah, I see the ectogenesis stuff is right. I mean, that's another thing.
I'm a big fan of sci-fi in general, and I know I have no problem and discussing, but we have to be clear about what are we exactly talking about.
Absolutely, and I'm glad you put that out there, because we did an episode on sort of artificial womb technology, which for the end of the process, and they too brought up the whole this isn't ectogenesis either. So I'm with you on that. One of the things I wanted you brought to mind, because as you're talking about sort of dissecting these black boxes that exist, I mean. And again, sorry, take us back in our time machine here for a moment, but obviously one of the things when we look back into the early 1970s and some of that original work by Beatrice Mintz here in Philly, specifically the in vivo teratocarcinoma reversion in those chimeric models, that's another sort of area that and I think about it like embryos are very good at absorbing junk or bad stuff and organizing out what they don't need. Yeah. And whether it's the Mintz embryo work, which in cancer is still sort of a black box today of how that all worked, or some of the other work in that area, like throwing in somatic tissues into embryos, viruses, all this other stuff, very good at getting rid of a lot of stuff. Any thoughts on sort of another component of renewal bio, looking at sort of relearning or looking back at sort of the 50 years of history here, and what's going on in some of that, because this is very important biology as well.
Yeah. I would say it's more, I would say that would be more my lab than renewal bio. I think renewal bio in the sense like that's a company which I'm advisor, by the way, I'm not chief scientific officer. They're very much focused on the humans, trying to get cell types and improve those and characterize them. But, of course, I think along the way, they will develop a lot of technologies which could be useful for many, many other things. Yeah. So, I'm very happy about this exchange, because I will, my lab, which is typically, looking at more, I call it the geeky stuff, but like what you mentioned. I think there are a lot of, it's funny that you mentioned Beatrice Mintz and also, like Rosa Beddington, also like a lot of, it's been fascinating for me, and trying to dig deeper even in some of the papers from the 70s, and because there's been like really cool stuff that's been done. For example, just to give you an example, like Beatrice Mintz actually showed that very, very inefficiently, but actually you can take an embryo at day 7.5. So you cannot put it back into a uterus on itself, but actually if you bring it back into a female that is already pregnant, and you open up the pregnancy sac and you just stick it in addition to the existing embryo, at 5%, they actually somehow survive to only day 15. So we have to really, it's been really, a lot of really these giants have done some really careful work, very technically challenging. It's very important to look at. And, because, in a way we're modernizing what they're doing and kind of perhaps even taking different pieces and just putting them together again and looking at this. And as I said, like a lot of like interesting questions that, as you went, just to kind of give you more examples, I think for example, these systems also allow us more easily to do experiments where, let's say you want to actually make a mutation only in the placenta, but not in the embryo, or make a mutation only in the yolk sac. So you're starting having actually these games
Yep.
Or actually, if you think about what happens if you mix, I don't want to talk about humans, actually, monkey a mouse, or even rabbit and mouse. Was I told you the mouse embryo, the embryo has like this egg cylinder shape versus the rabbit has a bilaminar disc. So what is dictating the shape? Is it the placenta that is making forces? And then you will then, if you actually mix mouse placental cells with a rabbit ES cells, is it going to be a bilaminar disc, or is it going to be an egg cylinder shape? What is there are a lot of course, there are a lot of limitations to these experiments, but you can start thinking about these experiments, which also you couldn't do before, or they were done, but in much limiting capacities back in the 70s. And so that is a lot. Definitely, I agree with you. I think there's a lot of the questions to look at it. And then the other thing, which is also the fact that we can grow the embryos outside the uterus now, I think you start to really take to the next level two things. One, which is mechanobiology of the embryo, because you can much more control the mechanics and precanic pressure than when you look inside the uterus. And other thing which you couldn't do is optogenetics. As you know, optogenetics is a field that has revolutionized brain neuroscience, but you couldn't really do optogenetics inside the uterus. But now we have the embryos growing in glass bottles, so they can expose them to light. So you can now think of next level of genetic engineering by optogenetics and really control cell types by light on off to stop their migrations. And really something we're thinking about. And again, some old ideas, maybe now they can be brought into reality to some extent. So we'll see. And as I said, I think definitely my lab is gonna be very busy about this, and I think there's the field in general is really gonna boom more.
Yeah, I mean, you definitely will be busy, and as I said, as you said, the remarrying of some of these tools finally in a structured way with these technologies that you developed. I clearly think that you're ushering in a whole new era of this, and as I said, I mean, it's been fascinating, and you clearly are the next step in this amazing lineage that's been developing through these thought leaders. And I'm going to be rooting you on. I, for one, just as you continue to develop, both at the lab and Renewal Bio. So really, really very impressive. Jakob, before I let you go, anything else coming up for 2022? I know you have some papers coming out, I guess, but any other conferences, any place that will run into you, maybe talk to you, meet you on the sort of regenerative biology circuit?
Well, I did not mean to advertise, but we are actually having a very nice early embryo epigenetics conference here at the Weizmann. We call it EEE, Early Embryo Epigenetics, in February. Cool. And of course, it's a we're bringing really all the leaders in the field. I think it's going to be really fun. And Israel actually is very warm in February, so it's open.
There you go.
I think, by this one and the other meetings, I think, it will take us some time on some of the progress that we're making, because as I mentioned, we modestly speaking, I think these kind of experiments, we take our time. We really want to get a lot of numbers of embryo, test many conditions, really characterize them. So it a little bit takes time to report the progress, but I think we're a lot of encouraging, as I mentioned, direction. We have positive results on growing rabbit embryos, about getting human synthetic embryo models as well, very early stages, and we characterize them. It will take us time, and I think but this is progressing, and we'll always try to report this as soon as possible once we feel we have solid data and complete. But I think we're very encouraged by this, because as a senior, definitely we already know that this is not a species specific phenomenon, and of course we have failures as well, but it's exciting, and I think we're really happy that other groups are also starting to use this, and because I think there's so much to be done and people have different ideas and different ways of doing things. So I think really hope and I believe that they will have very important input from others as well that are starting with similar platform or the same platform.
Outstanding. Again, for everybody that's going to be listening to this episode of the podcast across the various networks or watching on the YouTube channel, again, you've been listening to Dr. Jakob Hanna, Senior Scientist, Professor in the Department of Molecular Genetics, Weizmann Institute of Science in Israel. Check out his lab, check out his new company, Renewal Bio. Jakob, I want to thank you again for taking the time out of your schedule to come talk to us for a little while about all these themes. Obviously, thank you for everything you're doing there. And as we like to say on our show, thanks for helping to create a better tomorrow via what you're doing. It's really an amazing set of programs. I'm going to be rooting you on.
Thank you. It's really my pleasure. And, thank you for covering this topic. We really want people to understand what we are really doing and be transparent about our thoughts and the slides of it. It's everything is very engaged, both the scientific community and the general public.
Absolutely. Be well.